Today we will discuss about cheese starter cultures. Cheese is a dairy product that is produced by coagulation of the milk proteins. The milk is usually acidified using the lactic acid producing bacteria and coagulated by adding the enzyme rennet. The solids are separated, further processed and pressed into final form. Calf rennet or microbial rennet and recombinant rennet are now in use. Previously only calf rennet was available, which was extracted from the stomach of the calves. However, now it is microbial rennets and other alternatives are available. Cloned rennet is also used in the industry. There are different types of cheeses according to the different processes which are used for making cheese. Unripened or fresh cheeses are there, which can be consumed directly after preparations, whereas ripened cheeses are kept stored for a longer period to allow aging or different kind of proteolytic and lipolytic changes. Ripening can be internal ripening, which is through bacterial action, or it can be surface ripening, that is smear ripening. And sometimes in specific varieties, the molds are used for ripening of cheese, such as blue veined cheese, and these are known as mold ripened cheeses. Milk coagulation is one of the important aspects. Rennet or the enzyme that sets the cheese into strong and rubbery gel. The curds produced by acidic coagulation alone are rather fragile. So, that is why action of rennet becomes very important. It enables curdling at a lower acidity. Important because flavor producing bacteria can be inhibited in the environment having higher acidity. So, that is why rennet acts early and the pH is still higher as compared to acid coagulation. In milk, softer, smaller, fresher cheeses are curdled with direct acidification or using more of acid than use of rennet, whereas harder varieties of cheese, that is larger, longer aged varieties, are mainly curdled by rennet and acid production is lower in the beginning. The formal definition of cheese starter culture, if we see, the starter bacteria could be defined as lactic bacteria strains which produce sufficient acid to reduce the pH of milk to 5.3 in 6 hours at 30 to 37 degrees centigrade. Starter bacteria are deliberately added to milk at the beginning of the manufacturing process. Many artisanal cheese varieties were made from raw milk using natural contaminants of the milk as starter organisms. They grow during manufacturing and attain densities around 10 to the power 7 to 10 to the power 8 colony forming units per gram within hours of the beginning of the manufacturing process. However, these artisanal cultures are not very consistent, and it is the environment which becomes very important, that is temperature and other controls. Cheese starter cultures according to the optimal growth temperature are categorized into either mesophilic cultures which love to grow between 25 to 30 degrees centigrade, or thermophilic starter cultures which are used in the range of 35 to 40 degrees centigrade, and the thermophilic are also used where cooking temperature, higher cooking temperatures are used for the curd, and this depends on the type of cheese to be produced. So, we normally assign two types of functions, that is primary functions of the starter culture and secondary functions of the starter cultures. So, in the primary functions, acid production is the most important, then flavor production, eye formation. Sometimes in typical varieties of cheese, eye formation becomes very important, like Swiss cheese or Dutch varieties of cheeses, where specific bacteria are used for gas production, and these holes appear as eyes in the cheese. Inhibition of undesirable microorganisms, that is due to the acid production, proper rate of acid production and faster lowering of the pH. The spilage organisms and also the pathogenic organisms are inhibited, they do not proliferate in the milk, thus ensuring the safety of the product. Secondary functions include different activities like particular varieties where we need surface growth, or we need blue veined cheese and softening of the texture of the curd. So, in all these parameters, acid production is the most primary activity, and milk is preferred medium for the growth of bacteria because it has sugar available, and this lactose sugar is the major sugar which is fermented to produce lactic acid. The culture inoculates the milk with the desirable type of bacteria, proliferate for consuming the milk sugar and converting this into lactic acid. You can see the lactose is a disaccharide which is broken down to glucose and galactose, and these two sugars, hexoses, glucose and galactose, are converted to lactic acid by further fermentation through the Entner-Meyerhof-Parsonage EMP pathway. In this process, lactose is converted to lactic acid and thus raising the acidity and lowering the pH, which assists in the coagulation of the milk proteins by the action of rennet or by developed acidity itself. The developed acidity also prevents the spoilage and harmful bacteria from gaining a foothold right from the beginning. Mesophilic cultures, as I told you, they have the temperature of around 25 to 30 degrees centigrade for optimal growth, and they are used for production of cheddar cheese or Gouda cheese, Edam, Blue and Camembert cheese. These are mainly semi-hard varieties of cheese. In mesophilic DL starters, there are aroma producing organisms also, and they produce because Leuconostoc is added along with Lactococcus lactis subspecies cremoris or Lactococcus lactis subspecies lactis. The Leuconostocs produce flavor and also produce gas. Mesophilic DL starters commonly comprise an undefined mixture of strains of these species as mentioned earlier. The thermophilic cultures are used for high temperatures cooking, that is hard varieties of cheeses such as Emmental, Gruyere, Parmesan and Grana, and comprises of Lactobacillus species, Streptococcus thermophilus and Enterococcus species. According to the industrial classification of cultures, the most frequently used cultures are mixed strain cultures, which contain two or more strains of bacteria which can support each other in their functioning, and single strain cultures are also used, which are defined strain cultures. Mixed strain cultures are a cocktail of mesophilic bacteria or thermophilic bacteria, or sometimes a combination of both. These cultures not only produce lactic acid, but also have the ability to form gas and aroma components. Carbon dioxide is essential for creating the holes in round eyed cheeses and supports the openness of granular type of cheese. Gouda, Manchego, Tilsiter are based on mesophilic cultures and Emmental and Gruyere on thermophilic cultures, which also have propionibacteria for production of large amounts of carbon dioxide and the typical nutty flavor. Single strain cultures are mainly used where the object is to develop acid and contribute to protein degradation, that is in cheddar and related types of cheeses. These defined mesophilic cultures are now commonly used for the manufacture of most cheddar cheese in the world. Three characteristic abilities of the starter cultures are of primary importance in cheese making, that is production of lactic acid, and they break down the protein and when applicable also produce carbon dioxide. The main task of the culture is to develop acid in the curd, and when milk coagulates, bacterial cells are concentrated in the coagulum. Formation of acid lowers the pH, which is important in assisting synresis, that is shrinking of the curd or contraction of the coagulum accompanied by expulsion of the whey, so that the appropriate moisture is obtained in the product. Salts of calcium and phosphorus are released, which influence the consistency of the cheese and help to increase the firmness of the curd. Another important function performed by the acid producing bacteria is to suppress the growth of contaminating bacteria and heat resistant bacteria that survive pasteurization. Production of lactic acid stops when all the lactose in the cheese has been fermented or consumed. Lactic acid fermentation is normally a relatively fast process. In case of cheddar, it must be completed before the cheese is pressed, and for other types within a week. If the starter also contains CO2 forming bacteria, acidification of the curd is accomplished by production of carbon dioxide through the action of citric acid fermenting bacteria. Heterofermentative bacteria in a mixed strain starter culture have the ability to develop CO2 and are essential for production of cheese with a texture of round holes or eyes. The eye formation is so important in Swiss varieties of cheeses and Dutch varieties of cheeses. The evolved gas is initially dissolved in the moisture phase of the cheese, and when the solution becomes saturated, the gas is released and creates the eyes. There are several factors which affect the performance of the cultures. These are known as disturbances in the culture, and the culture becomes either slow or totally stops the production of acid, which we term as a dead vat. So, these disturbances can be due to several reasons, and one of the most common causes is the presence of inhibitory residues in the milk. When the milk is drawn from the animals which are being treated for antibiotics, the antibiotic is excreted or it comes down in the milk. So, such milk becomes inhibitory to the growth of these starter organisms, and either acid production will be slow or it will be totally stopped. So, milk from treated animals should not be used for making any fermented products, especially cheese. There are several recommendations that milk from antibiotic administered animals should be withheld after the last injection or last dose of antibiotic has been given for another 4 or 5 days. Normally, the milk is pooled to dilute the effect of inhibitory residues, and pooling dilutes the concentration of inhibitory residues. There are tests available to detect the presence of the inhibitory substances in milk, and if the milk is pre-tested for the absence of these antibiotics residues, then such milk is to be preferred for the preparation of the cheese. Cultures also are inhibited if there is a temperature shock. If so, the temperature should be controlled properly in the vat, and they should not be allowed to go higher. Otherwise, the culture will get a shock, temperature shock, and the final acidity will not be attained in the optimal time. Similarly, there are other inhibitory substances like we have bacteriophages problem. Bacteriophages are heat stable viruses which are parasites on the starter bacteria, and they infect the starter organisms and also multiply inside the starter cells by killing the starter bacteria. Their proliferation rate is very high. We call this as the burst size. 32 or 64 or 128 daughter viral particles are produced in one cycle from one cell, and this rate surpasses the rate of bacterial growth. So, that the culture is sensitive, all this, it is a single strain culture, then it will be inhibited totally. Or if it is a paired culture, that is having 2 strains, then only one strain may be resistant to the bacteriophage. So, that the acidity will continue, but it will be slow. In mixed strain starters, they are selected for different phage types, and these phage types permit us the continued activity of the starter cultures, because all are not inhibited by the bacteriophage. So, the phage inhibition can be prevented by rotating the starters cultures in the cheese plant, so that the buildup of the phage does not take place against a particular strain, and the milk should be treated cheese milk should be treated at more than 80° centigrade for minimum of 15 minutes, so that the fudges in the raw milk are totally killed. The general sanitary conditions in the plant should be maintained, and air pressure, positive air pressure should be maintained in the cheese room. Using HEPA filters, the filtered air should be allowed inside, whereas no direct air should be allowed inside the cheese room where the cheese is being prepared. The starter cultures are also important to be chosen in that way, so that the bacteriophage, they are resistant to the bacteriophage. Many lactococci are susceptible to phage attack, and they are divided into different phage groups, and accordingly the paired cultures or multiple strain cultures are prepared. So, beforehand the commercial starter culture company can give the information about the status of the phage sensitivity of the cultures, and if phage develops against a particular culture, its use in the plant should be totally withdrawn, and such precautions should be taken. Another disturbance in the culture is due to the presence of sanitizers. We use chemical sanitizers in the plant for cleaning the dairy surfaces of the equipment and other vessels, and these residues of sanitizers are inhibitory to bacteria in general, though they normally do not result in dead wet, they can result into slow wet, and if the acid production is slowed down, the cheese quality will be compromised, because it will not suppress the growth of the the undesirable bacteria, and these undesirable bacteria can multiply further and leading to microbiological spoilage of the product. We do not get the kind of flavor and texture and taste which we are looking for. So, starter cultures are very important to have good quality of the cheese, and you can prepare these starter cultures in the dairy itself by making your own bulk culture, or you can prepare directly purchase the direct wet starter cultures from the industry. There are starter culture suppliers who will give the freeze dried cultures or frozen concentrated cultures, which can be directly used. Or if you have your own culture facility, you can raise the bulk culture yourself using all the precautions and making sure that the final pH or final acidity is not over acidic, because exposure of the cultures to higher acidity compromises with the activity of these cultures, makes them slow, and thus the acid production rate comes down. So, starter cultures of particular type are to be used, and secondary starter cultures like Propionibacteria bacteria or Penicillium camemberti or Brevibacterium lineans are used to be in the appropriate manner for specialty cheeses. They are the ripening of these cheese varieties, specialty cheese varieties requires special conditions, and that can be met in the industry, and use of appropriate cultures will go a long way in ensuring the quality of the final product which we obtained. So, this is the general introduction to dairy starter cultures, cheese starter cultures. Thank you.